Hello everyone, welcome to Dr. K. Prem lecture series. In today's lecture, we'll discuss about end labeling. This is one of the method or way to label the DNA molecule. In general, there are two ways of there are two ways to label the DNA molecule. One is end labeling, and the second one is all along the molecule. There are two ways. One is end labeling, and the second one is all along the molecule. So, in the today's lecture, we'll focus on end labeling. End labeling means here also in end labeling you can do labeling at 5' site or 5' site or 3' OH. In the 5' site where 5' phosphate is labeled with the replaced normal 5' phosphate is replaced with the radioactive phosphate, that is called as a 5 prime phosphate labeling. And the second one is 3 prime labeling. 3 prime labeling means labeled nucleotides. Labeled nucleotides are incorporated at 3 prime end of DNA fragment, that is called 3 prime labeling. So now we will move more detail of 5 prime labeling and 3 prime labeling. You see, 5 prime phosphate groups of DNA fragments replaced with the radioactive terminal gamma phosphate of ATP. 5 prime phosphate group of DNA, 5 prime phosphate groups of DNA fragments are replaced with the radioactive terminal gamma phosphate of ATP. So, as you all aware that this replacement is done by the a protein molecule or enzyme molecule is called as a polynucleotide kinase, PNK. Polynucleotide kinase catalyzes the transfer of terminal gamma phosphate group from ATP, the gamma labeled ATP, to 5' OH, 5' OH groups of polynucleotides or DNA fragments or oligonucleotides by forward reaction. This is a 5' phosphate label. The second one is 3' end labeling. 3' end labeling. In 3' end labeling, the alpha phosphate labeled ATP are used to label the 3' OH of 4 DNA fragments. This reaction is catalyzed by terminal deoxynucleotide transferase. Terminal deoxynucleotide transferase. There are two enzymes for this one end for end labeling. 5' phosphate end labeling is by polynucleotide kinase. 3' end labeling done by terminal deoxynucleotide transferase. And here for 5' phosphate labeling, radioactive gamma phosphate is transferred from ATP to 5' OH of DNA fragments. But in case of 3' labeling, alpha phosphate group labeled ATP. This is the difference between the 5' phosphate labeling and 3' end labeling. So the alpha phosphate is labeled in case of 3' end, gamma phosphate is labeled with the labeled in case of 5' end labeling. You see here This is what is a flow chart for labeling of piperin phosphates. DNA fragments are treated with the alkaline phosphatase. Alkaline phosphatase is an enzyme which dephosphorylates the removes the phosphate group by breakdown the phosphomonoester bond. So alkaline phosphatase removes the 5' phosphate and forms the 5' OH. When it is removed, whatever is there, what will be there at the ribose sugar, that will be 5' OH. So dephosphorylated DNA fragments with the 5' OH, dephosphorylated DNA fragments with 5' OH incubated with the PNK and radioactive gamma phosphate labeled ATP. There are three phosphates, alpha, beta, and gamma. The gamma is terminal phosphate. The beta is in between, and the alpha is attached to the alpha phosphate is attached to the ribose sugar, right? So PNK has ability to transfer the terminal phosphate group of ATP to 5' OH of DNA fragments. Hence, gamma phosphate labeled ATPs are used here, right? And the PNK performs the forward reaction and phosphorylates the 5' OH DNA termini. Then the probes are ready. And the probes are very, very specific. And in signal may be less or signal may be lesser, but they are very specific. Means there is no addition of nucleotide. These probes can bind with the its own target because we did not add any nucleotide to the probe. They'll go and bind and they'll hybridize with the only respective molecule. So the 5' labeling provides the more specific probes and less signal. And now we will see the same thing in the diagrammatic representation. You see, here, these are the single strand DNA molecules which are having the 5' phosphates and 3' OH. They are treated with the alkaline phosphatase, which can cleave the phosphomonoester bonds and forms the 5' OH. So this alkaline phosphatase can cleave removes the phosphate groups from 5', and you see the phosphate is removed, and the resulted DNA fragments have 5' OH and 5' OHs. Now these DNA fragments, in dephosphorylated DNA fragments, are treated with the T4PNK, that is polynucleotide kinase, and they're incubated with the gamma labeled phosphate of ATP. So T4PNK, that's a polynucleotide kinase can catalyze the transfer of gamma phosphate, that's a terminal phosphate of ATP, to 5' OH of dephosphorylated fragments, and then at the result of this reaction, you see phosphorylated or labeled phosphate groups are at 5' end. These are called as probes. So PNK preferentially labels the protruding ends, means orangs, but not the recessed one. And but if you can include the PEG, polyethylene glycol, in the reaction condition, it can phosphorylate the even blunt ends also. The most important molecule in this reaction is ATP molecule. You see, this is what is the ATP molecule, where alpha phosphate, beta phosphate, and gamma phosphate. For this reaction to 5' phosphate labeling, the gamma phosphate is radioactive. Gamma phosphate is radioactive by, you see, radionucleotide, where gamma phosphate is radio labeled. So this radio labeled gamma phosphate is transferred to the 5' phosphates or 5' ends of the DNA fragments. 5' DNA fragments. This is how you can label the 5' phosphate groups, and this is nothing but 5' phosphate labeling. Next one is 3' end labeling by TDT, that's a terminal deoxynucleotide transferase. Before going to the reaction, I want to explain the ATP molecule you use here. You see, you know that ATP is having three phosphates, alpha, beta, and gamma. But here, alpha labeled phosphate is most important. Alpha labeled P32 means this group is labeled with the radioactive phosphate. Why? Because this molecule go and attacks the 3' OH and removes this gamma and beta phosphate as a pyrophosphate, and alpha phosphate of nucleotide is involved in the phosphodiester bond formation. Which molecule of which phosphate molecule of nucleotides are involved in the phosphodiester bond means alpha phosphate molecule, alpha phosphate of nucleotides are involved in the phosphodiester bond formation. So, a 3' end labeling, uh, alpha phosphate is labeled, and once the uh, polymerase catalyzes the reaction and the alpha phosphate is involved in the phosphodiester bond formation, and then there is a release of diphosphate. They are nothing but gamma and beta, right? Same thing I want to tell you here. DNA fragments are incubated with the terminal deoxynucleotide transferase and radioactive. What phosphate is? Alpha phosphate is labeled ATP. Now, the terminal deoxynucleotide transferase catalyzes the incorporation of alpha phosphate ATP at 3' end of DNA fragment in a template independent manner. So the 3' OH is it when it is labeled. Here we are using the terminal deoxynucleotide transferase, which can incorporate the nucleotide at 3' end without need of template, right? So it will add the nucleotide at 3' end, and that is if it is at labeled and 3' end is labeled. Probes are ready with the labeled nucleotides at 3' OH. The same thing you can see in the diagrammatic representation. You see these are the DNA fragments, and they are treated with the TDT and alpha label 32P. Here alpha phosphate is radioactive. Now when these two components are there along with the DNA fragments, and this alpha labeled ATP is incorporated into the DNA fragments by the action of TDD, and then the probes are prepared. You see, probes are more sensitive. These probes are more sensitive, and 3' end labeled probes are more sensitive and less specific. They are more sensitive and less specific because you are adding in nucleotides to the 3' end. They are not part of the target. They are not part of the target, but they are very sensitive because you are adding more labeled nucleotides at 3' end. So you get the more signal, but they are very less specific. So TDT can incorporate more ATP and deoxynucleotide DATPs and DTPs instead of DC and DC. And so you can use the ATP or TTP to nucleotides to incorporate at the 3' end. So this is what is the end labeling, and you try to remember 2 points from here. Take home message, sir. 5' end labeling is more specific and less sensitive. and their gamma phosphates are labeled. And second one is 3' end labelings are more sensitive and less specific, and here alpha phosphate is labeled. Hope you understood this concept. Thank you all for listening. If at all you like it, just subscribe for regular updates. Thank you all. See you again. Bye bye.